So we are going to start talking about fatty acids, and we'll start by talking about how they're broken down. Here we're going to discuss just a general overview of fatty acids. So what we want to take from this overview is to know what basically lipids are doing, what's their role in our body, what the basic biochemistry of fatty acids are, what is their structure, how are they relevant. They're going to be important, of course, in our membranes, but they're also important in many different processes in the body. We're going to talk about omega-3 and omega-6 fatty acids and what they become, and also just know general structures of our triacylglycerols as well as phospholipids and our plasmalogens. Now, so fatty acids in the body are going to be made up of all the membranes, including the plasma membrane, the endoplasmic reticular membrane, Golgi apparatus, lysosomes, any other organelle inside of your cells. They're also going to be a major component of stored energy, and the stored energy is in the form of triacylglycerides, and they are in the adipose tissue. The nice thing about fat is that it is hydrophobic, and so it does not absorb a lot of water, and so you can store a lot of fat. They're also going to be made into what we refer to as bioactive lipids. And so these are lipids that can physically do things. Platelet activating factor, for example, is a bioactive lipid. So in essence, what is a fat? And there are several things to think about when you think about what fats are. And so your fats are going to be either saturated, and your saturated fats are going to be where there's no double bonds, and so this is a saturated fatty acid. They're also going to be long chains of hydrocarbons. So you have a lot of hydrocarbons that are going to be on a fat molecule. You're going to have the numbering of the fat starting at the end with the carboxylic group. So here's our carboxylic group, and so the numbering is going to start there. And what's important to understand is at a physiologic pH, the carboxylic group is going to be ionized, and therefore fatty acids have negative charges in biologic fluids. Now, of course, they have a very long hydrocarbon chain, so they're still going to be primarily hydrophobic. But it's important to understand that they do have that negative charge. All right, so saturated is no double bonds. And then, of course, unsaturated means they have double bonds. And so for the unsaturated, you can have polyunsaturated. And this, of course, means that they have multiple double bonds. Now, in general, the melting point of a fatty acid will increase as the number of carbons increase. So you increase the number of carbons, results in an increase in the melting point. Also, an increase in unsaturated sites decreases the melting point. So fatty acids of less than 8 carbons are typically liquid at a physiological temperature, whereas greater than 8 is a solid. So our fatty acids will have sometimes double bonds, and the double bonds are going to be designated by a delta sign. And so we've discussed this. And so we have here an 18 carbon. And so this is given an 18 for the number of carbons. It has a number 1 next to this, and this is denoting it has one double bond. And then it's telling you where the bond is because it has the delta 9, so you know that the double bond is going to start at position 9. So the delta bond is between carbons 9 and 10 in this 18 carbon molecule. And then down here we have a polyunsaturated fatty acid. That's a 20 carbon in length. It's going to have 5 double bonds, and the double bonds are going to be listed here. They start at positions 5, 8, 11, 14, and 17. So a double bond is between position 5 and 6, 8 and 9, 11, 12, 14, 15, 17, 18, for example. And this is actually an omega-3 fatty acid that's important in our diet. It's eicosapentaenoic acid, or EPA. So long-chain fatty acids are going to be very common in our diets, and we actually make long-chain fatty acids. We primarily are going to make in our liver palmitate, carbon-16, and then from that we can make longer fatty acids. We can build upon that. In our diet, we're going to eat palmitate, stearate, and other oleate, linoleate. And all of these are going to be on our diets. Now, in nomenclature, the omega starts at the opposite end of the carboxylate. And so when you're talking about omega fatty acids, they get their number or their name, omega-3, omega-9, based on where their first double bond is. So at the omega end, of course, you're going to have the double bond at 1, 2, I should make it a different color. This is at the omega end, 1, 2, and 3. So the omega is an omega-3. The omega-9 is going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, where that double bond starts at the 9 position, so it's called an omega-9. And so this is the same nomenclature. You don't change the molecule, you're just starting the counting from the opposite end, and that's referred to as the omega end. All right, so we're going to start talking about how we break down fatty acids or how we build up fatty acids. And so there's going to be a lot of enzymes, and the enzymes are going to be specific for how long the actual fat is. And so these acyl-CoA synthetases, for example, are going to be specific for different lengths of your fatty acids. Now, a very long-chain fatty acid, which is in general more than 20 carbons, can only be oxidized in your peroxisomes. Most of our fats are going to be long-chain fatty acids, so this is a majority. Very long-chain fatty acids are important in the brain, for example. But most of our body is going to use long-chain fatty acids as energy sources because that's what's stored primarily in your adipose tissue. And so these are going to be oxidized at different locations. Membranes of the endoplasmic reticulum, which is our omega oxidation, mitochondria, our beta oxidation, and also in peroxisomes. So long-chain fatty acids can also be metabolized in peroxisomes as well. Medium-chain fatty acids are different. Medium-chain fatty acids and our acetyl groups are going to be used up directly. They never get stored. So they are going to go to the matrix of the kidney or the liver, and the acetyl group is going to hang out in the cytoplasm and be used as a building block, or it can be oxidized in the mitochondria matrix. And so what's key here is you can store long-chain fatty acids. Oops, sorry, I don't want to do that. And under this line, you cannot store. So if you have a lot of medium-chain fatty acids in your diet, they're going to be oxidized immediately. They're not going to be stored. All right, so now a little bit about our famous omegas. We have omega-3 and omega-6. So omega-3 and omega-6 come from plants. And so omega-3 important is alpha linolenic acid. And this is going to become two important anti-inflammatory active lipids, eicosapentaenoic acid and dohexanoic acid, EPA and DHA. Now, all you have to do is eat plants and your body will make these. It has no problem making EPA and DHA. Your fish can also be high in EPA and DHA, and by eating the fish, you're going to skip those reactions to generate those. Now, another is going to be your omega-6 or linoleic acid. This is important from plants again. You can get it from vegetable oil as an example. This is going to be converted eventually to your arachidonic acid, and it has several steps to get there. Arachidonic acid is going to be inserted. So arachidonic acid is going to be primarily taken from position 2 of your membrane phospholipids. So your dihomogamma-linolenic acid is inserted into that membrane position so that when your cells need to make arachidonic acid and any of its biologically active lipids, they can simply just take it from position 2 in the cell membrane. So it's kind of a stored place for a biologically active lipid. All right, let's just talk a little bit about basic structures. We've seen tags before. We have our glycerol backbone, and then we have our R1, R2, and R3. And these are all going to be fats. They can be different lengths of fats. Again, in position 2 is going to be a precursor for arachidonic acid primarily. So R2, if it's in the membrane, can be that D-gala arachidonic acid precursor. We have phospholipids. And phospholipids are, of course, going to have at that 3 position a phosphate group, and then it's going to have something attached to that phosphate group, some R group. And so phospholipids that are important in our membranes are phosphatidylethylamine, phosphatidylserine, on the inner leaflet, phosphatidylinositol, inner leaflet, etc. And then finally, our basic structure of plasmalogen. Now, plasmalogen is going to also have a phosphate group, and it's going to be an important biologically active lipid. One of the more important plasmalogens is going to be platelet activating factor, PAF. And so in platelet activating factor, it's a little bit different because now not only in carbon 3 do you have the phosphate and then your R group, but also on carbon 2, you're going to have an acetyl group attached, and so you don't have that long chain fat. So in a platelet activating factor, you only have one long chain fatty acid. And so this is a highly active lipid.